Hi everyone, welcome to my channel. Today we will learn about BLAST tools. BLAST is defined as a comparison of the query sequence with sequence database, and it finds the similarity between the query sequence and sequence database. So BLAST is abbreviated as Basic Local Alignment Search Tools. BLAST is divided into 5 types: BLAST-N, BLAST-P, BLAST-X, T-BLAST-N and T-BLAST-X. So what is the work of BLAST-N? It compares the nucleotide sequence to the nucleotide query sequence or a database of a nucleotide sequence. The both sequence or the nucleotide based sequence. Next one is BLAST-P. Here one or more protein query sequence to the subject protein sequence or database of the protein sequence has been compared. So both of the sequence are taken as the protein query sequence. Next, BLAST-X. BLAST-X is compare the nucleotide query sequence against the database of the protein sequence. Here both nucleotide and protein are compared. Next, T-BLAST-N. Compare protein query sequence against seq frame translation of database of the nucleotide sequence. Here also the both protein and nucleotide sequence are compared. T-BLAST-X. Compare translated nucleotide query sequence against translated nucleotide sequence database. Here the both are nucleotide, but the difference is that nucleotide query sequence is against the translated nucleotide sequence database. Next, we are going to see about the applications of the BLAST. The first one is homology searching. It is a fundamental technique in computational biology used to identify the similarities between the DNA, RNA and protein. Second one is species identification. We can identify the exact species of an organism based on their morphology, genetic and molecular features. Third one is phytogenetic relationship. It means reconstructing the evolutionary history and relatedness among the species, genes and protein. DNA mapping and comparison. It is one of an essential technique in bioinformatics for structure variation and evolutionary relationship. See how to use the BLAST tool. So first I will go to the Chrome and I will look for an NCBI. So this is an NCBI platform, so National Center for Biotechnology Information. We need to click this. So this is the interface. Okay. Now here in a popular resource, there is a BLAST. So this is the page of the BLAST. They have given that basic local alignment search tool, and the definition is that region of similarity between the biological sequence. It compares nucleotide and protein sequence to the sequence database and calculate the statistical significance. So here the nucleotide BLAST and protein BLAST. BLAST X means translated nucleotide is converted to protein, and T-BLAST-N means protein is converted to translated nucleotide. And here there are different types of blast like smart blast, primer blast. Use primer specific to MCR template and global align. Compare two sequence across their entire span. CD search. CD search means conserved domain in your sequence. And IG blast is specifically used for the immunology and T cell receptor sequence. And these are the multiple types of blast. So I will go for a nucleotide blast. So this is the page of the nucleotide blast. Here the different forms of blast are given like blast N, blast P, blast X, T blast N and T blast X. I have given the clear explanation about this in starting of the video. Next, here is the enter your sequence. We can enter a accession number of a particular sequence, or we can use FASTA sequence. So here I will use the FASTA sequence. So for taking the FASTA sequence, I will again open the NCBI website. Here is the, I will choose all databases. Here I will choose nucleotide. We can use any of the name. I will use insulin. Here I will choose the first one. You can choose any of the datas. Okay, so I have taken this insulin mRNA. Here is the FASTA file. Always the FASTA file is indicated by greater than symbol and some accession number and the title and the sequence. So we need to copy this to paste in the blast. So I will paste here. So if you have the FASTA sequence, you can copy and paste here, or you have a file form, you can upload the file form. Next, job title, we can use whatever the job title. So I choose here the name given. Okay, what is the sequence of the name? I will choose the same name as a job title. So here is a database or standard database we can use, or RNA, genomic plus transcript DNAs, beta coronavirus and experimental database. So these two are the recent invention. Okay, beta coronavirus and experimental database. Next, here they have chosen the core nucleotide database. Okay, and organism, you can include some other organism to compare with your FASTA sequence. Next, there is an exclude what you don't want, like models and uncultured experimental sample sequence. If you need, you can choose. If you no need, you don't want to choose. And limit to miss sequence from type material, which type of material you have taken, and that is given. And here is a program selection, like highly similar sequence. If you need this sequence to be highly similar, you can choose, or dissimilar, more dissimilar sequence and somewhat similar sequence you need, you can choose any of these. And here is the blast. We going to blast the sequence. So this is the result page. It takes some times to give the result. So this is the final result page. We have taken the job title was what I have mentioned in that. The same job title is given, and RID number and program Blastian, database core NT, query ID. This is unique ID for every sequence, and description and molecular type. What is the molecule type? Is DNA. Okay, and query length is given. 432 query length is reported. So there are four forms like description, graphic summary, alignment and taxonomy. Here first in description they have given many sequence of species and scientific name of the sequence and their maximum score. Maximum score means how much they have obtained, and total score and query cover. Query cover like 100%, 99%, 91%. They have given E value is 0, 7, E18, 1E, 3E. They have given this E value must be lower to give a better result. E value always must be lower. And they have given percentage, 100%, 81%. This compares the sequence, and they shows that how much percentage it has obtained. So like 100%, here see they have 74%, 72 and 70% also observed. And accession number is unique for unique sequence. Next we are going to see about the graphic summary. So graphic summary is that this is the query sequence what we have taken, and we compare the sequence with the query sequence. So two red lines indicate that they are equal to the query sequence. So red line and pink line and green line indicate different alignment score. If red indicates greater than or equal to 200, and pink indicate 80 to 200, and green indicate 50 to 80. So these are the query we need to compare. So first two lines are equal query, equal comparison is given. Pink shows greater difference, and green shows the equal differences to the pink. So these are the comparison with the query sequence and subject sequence. Next is alignment. In alignment, there are query sequence, and that is aligned to the subject sequence. So here they align equally, like G align with G, C align with C, A align with A. Here is the score, how much bits are there, and expect what we expected, and identities 100% is identified, and gap is like there is no gap in between the alignment. Okay, and here the stand is plus plus. If in query sequence it is 60, in subject sequence it is 60, so it is called as plus plus stand. Okay, if here see, here also plus plus, in this every alignment has the plus plus, so nothing is different. Okay, next we are going to see about the taxonomy. So taxonomy is that they have given organism, what are the organisms are present, and blast name they have given, and given the score for individual organism and blast name, and number of hits, how much hits they have, and the description for the organism. So we can compare organism to the in blast to see their similarities. Okay, so thank you.